Hello everyone. It is a great honor to present our team's latest research today. This study has been published in iMeta, and the title is Gut Microbiome in the Regulation of High-Altitude Adaptation. It is well recognized that the extreme environmental conditions at high altitudes, characterized by low temperatures, hypoxia, and intense ultraviolet radiation, are major factors limiting the growth and development of animals living in these regions. Previous studies on high-altitude adaptation have primarily focused on genetic and genomic aspects, whereas the mechanistic role of the gut microbiota in this process remains insufficiently understood. This study centers on host energy supply and investigates whether rumen microbiota rumen epithelial energy metabolism mediates the differential adaptive strategies of yaks and Holstein cows. Under high-altitude conditions, we selected yaks and Holstein cows that had lived at high altitude since birth as comparative models. A first experiment was conducted using a completely randomized design, with a total duration of 44 days, during which all animals were fed the same diet. First, multi-omics approaches were applied to compare differences in the rumen ecosystem between yaks and Holstein cows. In a second experiment, rumen fluid was transplanted from yaks to Holstein cows to verify the role of the rumen microbiota in high-altitude adaptation. Metagenomic sequencing of rumen fluid from 20 Holstein cows and 20 yaks revealed distinct bacterial and archaea communities between the groups. Yaks were enriched in 37 bacterial species, while Holstein cows had 54, including 21 Prevotella species that were significantly negatively correlated with rumen pH. Functionally, Holstein cows possessed more genes for complex carbohydrate degradation, whereas yaks showed lower carbohydrate degrading capacity but higher central carbon metabolism, highlighting species-specific rumen energy strategies. Metatranscriptome sequencing of rumen fluid from 10 Holstein cows and 10 yaks revealed clear separation of archaea and bacterial communities between the two groups by PCOA, consistent with metagenomic results. Functional correlation analysis indicated that carbon metabolism, central carbon metabolism, carbohydrate metabolism, and energy metabolism were positively associated with volatile fatty acid concentrations. Combined metagenomic and metatranscriptome results indicated that yaks favored central carbon metabolism, whereas Holstein cows were enriched in sugar-specific degradation pathways. More detailed schematic diagrams of metabolic pathways illustrated the core functional modules and key enzymes, as well as their expression patterns in the metagenomic and metatranscriptome datasets. Single-nucleus RNA sequencing yielded 45,682 rumen epithelial cells from yaks and 34,280 from Holstein cows, identifying 33 and 27 cell clusters, and 14 and 13 epithelial subtype S, respectively. Among them, yaks showed higher expression of genes involved in volatile fatty acid transport and ketone body metabolism, with pathway scoring confirming enhanced ketone body biosynthesis and fatty acid oxidation. In contrast, Holstein cows exhibited higher activity in VFA-cat-abolyse pathways. Furthermore, transplantation of yak rumen fluid confirmed the improved milk production and enhanced carbon metabolic functions of the rumen microbiota in high-altitude Holstein cows. In summary, this study elucidates high-altitude adaptation mechanisms from the perspective of the rumen ecosystem, clarifies the synergistic roles of the microbiota and epithelial cell functions in host energy metabolism, and provides new insights into leveraging gut microbes to regulate energy homeostasis in other animals and even humans under hypoxia conditions.